Hello and welcome to your Chapter 4 video lecture on the biology of the cell. We are going to start with section 4.1, which is an introduction to the cells, and here are your learning objectives. So first we want to talk about how cells are studied. In cytology, we are going to study cells by using microscopes, and they are measured in micrometers, but we are going to see that we have a deeper range in which we can see certain structures. So I put this illustration up to show you an idea of how these structures are found. You don't necessarily have to understand their math conversions, although I'm sure you are covering that in another course, but scientists are typically going to use micrometers. And we could see that we have the range of micrometers right over here, in which we can see our plant and animal cells, most bacteria, and chloroplasts, which are found in plant cells. And if we go above that, we have our millimeters. There are 1,000 micrometers in a single millimeter. And sometimes these micrometers are too large for scientists, so they have to measure down into nanometers, in which we can see things like viruses, proteins, and amino acids, and even atoms. And there are 1,000 nanometers in a single micrometer. So we use microscopy in order to view these small-scaled structures, and sometimes when we look at these structures, if we were to just look at the cells on their own, everything would look uniform, and it would be more difficult to see structures and tell them apart from one another. So we use staining techniques in order to provide contrast so we could see these structures more easily. And the types of microscopes that we have are light microscopes, which you've probably used in a lab before. These show us 2D images, and we pass light through the specimen in order to see it. Here, this specimen, which looks like some tissue taken from somewhere in our body, looks like maybe respiratory mucosa. It has been stained in order for us to see the cells. So you can see that the nuclei in here have been stained a darker purple color. We see the cell itself is stained this magenta pink, and then the cilia are a lighter pink. So we can make that distinction here, and then we can see that maybe we have a different type of tissue down here below it. Now next we have electron microscopes, and these are going to send a beam of electrons to illuminate the specimen and gives us greater magnification and resolution than a light microscope. So whereas a light microscope, we are able to see cells, but we have more difficulty seeing specific organelles. The nucleus is pretty much where we stop. Maybe we could see some mitochondria. But with the electron microscope, we can really see structures well. We could see a Golgi apparatus, we can see a rough ER, smooth ER, and so forth. And we have two different types of electron microscopes. We have our transmission electron microscope, which an image of it is right down here. We're using electron beams through a thin cut section, and we get 2D images. Whereas with our scanning electron microscope, which an image of it is right over here, we can direct that electron beam across the surface of a specimen and get 3D images. So here you could see the full scale of what cilia look like compared to this 2D image, in which we are just seeing almost like a cross-section of what a cilia looks like. And we saw that our cells are going to vary greatly in size and shape. So we can look at a erythrocyte, which is 7 to 8 micrometers, compared to a human oocyte, or the female's egg, that has a diameter of 120 micrometers. So you can see that we have quite a large scale change there, but most of our cells are going to be microscopic. And our cells are going to have different shapes. We have spherical, cube-like, column-like, cylindrical, disc-shaped, or irregular types of cells. And we see some of those in examples over here. So at the very top, we see that irregular shaped nerve cell in which we have the cell body over here and extensions extending on out of that cell body. We have the biconcave disc of our red blood cells, our cube-shaped cells, which can be found in our kidney tubules, our column-shaped cells, which can be found in our intestinal lining, and our spherical cells, like our cartilage cells, and lastly, our skeletal muscle cells, which are cylindrical in shape. Now let's look at the common features of our cell. So first we have our plasma membrane, which serves as our boundary, right? This is forming this outer almost fence-like structure, and it's limiting the barrier by separating the internal contents from the external environment. We do have some modified extensions here, such as cilia, which we saw in those electron microscope images, and also a flagellum, which would be longer here and gives motility to certain cells. And then we have microvilli, which you see over here, and microvilli really help with absorption and increasing surface area. Next, we have our nucleus, which is this structure over here. It is the largest in the cell, and it is enclosed by this structure, which is known as the nuclear envelope. You could see on our nuclear envelope, we have these pores or opening to allow certain structures into the nucleus and out. We're also going to see that our nucleus contains genetic material, such as DNA in our body. And this fluid that we have in here is called the nucleoplasm. Now, this darker spherical structure over here is called your nucleolus. Our nucleolus is going to assist with synthesizing ribosomes, which will help create proteins. So more on that in sections coming up. Now for our cytoplasm. Our cytoplasm can be found between the nucleus and the plasma membrane. So it's all of this in here. So it consists of our cytosol, which is the fluid in here, and we'll talk about that in the next slide, our organelles, and inclusions, which you see a couple inclusions here. We're going to talk more about them in the coming slides as well. So cytoplasm is all of this between the nucleus and the plasma membrane. Now for the cytosol. We said that that was the fluid portion in here in which we are suspending our organelles. We have a high water content in here, and it can also contain dissolved macromolecules such as glucose or proteins and ions. Now I mentioned that in our cytosol we are going to have our organelles, and our organelles really mean little organs. Each of these are complex and organized structures that have unique shapes and functions, and we can classify them into two categories. We have our membrane-bound organelles and our non-membrane bound organelles. So what does that mean? Our membrane-bound organelles, such as the mitochondria, are going to be enclosed by a membrane, and it helps to separate the contents from the cytosol that surrounds it. So this is going to include our endoplasmic reticulum, our Golgi apparatus, our lysosomes, peroxisomes, and the mitochondria that I mentioned earlier. Our non-membrane bound organelles don't have or are not enclosed with a membrane, and instead are composed of a protein, and we have examples of ribosomes. So you see some free ribosomes just floating in the cytosol here. Our cytoskeleton, which isn't really pictured here, I don't believe. They show just a few filaments over here. We've got three main filaments that make up the cytoskeleton, and it really kind of gives a framework to the cell. So more on that a bit later. We're also going to include our centrosome, which is pictured over here, and proteasomes. Now let's get back to those inclusions. So inclusions are in the cytosol. where it's going to be stored temporarily. So here are our inclusions. These are not considered organelles and instead are really just molecules added to and removed from continuously. So this can be things like pigments or the molecule glycogen, in which we can break down and obtain glucose from. And triglycerides are another example, which we can also break down and get energy from. What are the general functions of our cell? First, we want to maintain integrity and the shape of a cell. So this is going to be dependent on that plasma membrane and the internal contents. Another general function is to obtain nutrients and form chemical building blocks, and this helps us harvest energy for our survival. We also want to dispose of waste products. We are having chemical reactions taking place in our cell, and we want to avoid accumulation of that waste product. Otherwise, it can disrupt our cellular activities. And some are also capable of cell division. So we're able to make more cells of that same type and allow for new growth and help replace any dead cells or abnormal cells.